to indicate that you wish to ask a question. Okay.
Okay, can you, I cannot see you anymore, guys. Good morning, good afternoon, good night to everybody. I'm very excited to be here. Thank you, Shona, for the invitation. Can you hear me? Can you see me? Shona, everything is good?
Yep, we can see and hear you. Everything's working.
Okay. Again, I'm very excited. Thank you guys for coming to this seminar series. My lab is working on neurophysiology of mammalian hibernation, and we know about hibernation since Aristotle time. So he's the first one who described that when animals cannot escape unpleasant environment, when they don't have food and water, when the temperature is not pleasant, they will conceal themselves in underground burrow and they will stay there for a long period of time until they have resources available and temperature is pleasant. Despite that we know about hibernation since Aristotle time for such a long time, we still don't know a lot about cellular, molecular, and circuitry mechanisms that these animals employ in order to withstand prolonged period of food scarcity and low temperature. So hibernation is present almost in all mammalian clades in most subgroups. And here you can see examples of daily heterotherms, dormice, bats, shrews. And these are examples of, we call them true hibernators. And you can see here bears, our close relative dwarf-fat lemur. This is the first primate hibernator was identified. Our favorite animal model, ground squirrels. So despite that these animals, when you look at them, they look very different. When they hibernate, they undergo very similar physiological changes. And what's important here is that these changes will be lethal for us. And for these animals, this is part of the normal physiology. So specifically for ground squirrels, when they hibernate, they drop the metabolic rate by 95%. They also reduce the breathing rate, and when they're in deep torpor, they have only one breath per several minutes. They also reduce the heart rate from normal rodent heart rate 250, 350 beats per minute to only 3-4 times per minute. They also combine warm-blooded animal and cold-blooded animal in one animal. So when they're in active state, the body temperature is 37 degrees. They look like us. They have metabolic rate very similar to our metabolic rate. But during hibernation, they will drop their body temperature to ambient level. And because we keep our animals at the facility we call hibernaculum at 4 degrees Celsius, our ground squirrels, they will reduce the body temperature to 4 degrees Celsius, which is the temperature in your refrigerator. So again, they're going back and forth between being warm-blooded and cold-blooded animals. These animals can tolerate prolonged period of cold, and they can survive without food and water for up to 8 months. In my lab, we're working with 13-line ground squirrels. These animals, they obligate mammalian hibernators. What it means that they're on a very strong seasonal regulation. They will hibernate no matter what in the captivity at the end of August, the beginning of September. They occupy a very wide geographical range of North America, and specifically our ground squirrels are coming from University of Wisconsin at Oshkosh, from our collaborator, Dana Merriman. Without her, it would not be possible to perform our line of research. We have a long-standing collaboration already for more than 10 years, and several years ago, we also started to have our own breeding colony. So these animals, you can see they look like normal rodents, and they live in underground burrow. So seasonal cycle of 13-line ground squirrel actually includes several physiological states, and these are very important for some of you who are not familiar with this. I will just go through this because this is important for the rest of my talk. So during summer, when they're active, their body temperature is 37 degrees. They look like active rodents, and metabolically, they look very similar to us. At the end of August, the beginning of hibernation, our ground squirrels, they will reduce their body temperature to ambient level. And because we keep them in summer facility at 20 degrees Celsius, they will reduce it to 20 degrees Celsius. And this is already a sign for us that this animal is ready to hibernate. And we call this state pre-torpor. When we see that animal becomes hypothermic, we will put it in hibernaculum, and animal will hibernate at the temperature of 4 degrees, no food and water available. But when we talk about hibernation and obligatory hibernators, it actually includes two distinct physiological states. Torpor, that I just described, with reduced body temperature, reduction in metabolic rate, vital signs. But every 2-3 weeks, animal will wake up from torpor and will go into the state called interbout arousal, or IBA. During IBA, animal will increase body temperature to 37 degrees Celsius, will actually show a metabolic rate that looks like active-like, is not exactly like active state, it's something intermediate. And they will stay in the state for 24-48 hours. There are around 20-27 interbout arousals through the entire hibernation cycle. We don't know still physiological significance of interbout arousals. There are multiple hypotheses on this topic, but there are no fundamental studies, systematic studies to understand physiological significance of IBA. But this IBA is very important for hibernation maintenance, it looks like. So what's important here for the rest of my talk is that during the entire hibernation, they don't have food and water available. There are ecological studies when people actually demonstrated that there is no food and water available in underground burrow, but we also know in our facility that water is not available and food is not available for up to 8 months because we actually don't give them food and water for the duration of hibernation. And our animals feel just fine. What are the main challenges for hibernators? And these are currently main directions in my lab. Sorry. So our animals, they can withstand prolonged period of severe hypothermia and prolonged cold exposure. So what will happen to me if you put me in a refrigerator? So first I will start to develop cold-induced pain, then I will go into hypothermia, and then I will die. In case of hibernators, they don't die, they hibernate. And this is a fundamental difference between us and between animals that have ability to hibernate. And in my lab, we're trying to understand at the level of peripheral nervous system and central nervous system, how these animals adapt to cold, why they can survive prolonged period of hypothermia and cold exposure, and we cannot. It has a lot of therapeutical applications. I will not go through this, but we all can imagine that therapeutical hypothermia or cold allodynia, these are very hot topics in medicine. We're also working on seasonal reproduction, and Rafael Daipra, he started this direction in my lab several years ago. We know that these animals, they have ability to reproduce only once a year. They're on a very strong seasonal rhythms, and he's currently looking how metabolism is uncoupled from reproductive axis at the level of hypothalamic neurons. We're also working on hunger satiety regulation, and this is also very important in case of obligatory hibernators. They're going into this mode called seasonal anorexia, where they will refuse to eat for a long period of time for the duration of hibernation. And Sarah Moore, former graduate student in my lab, she recently identified some of the pathways that these animals fine-tune in order to go through reversible anorexia. We're also working on water deprivation and fluid ionic balance. So how do ground squirrels, how do hibernators can survive without water for a long period of time? What's happening with the fluid ionic balance during hibernation? Ni Feng, when she was a postdoc in my lab, and now she has her own lab in Wesleyan University, she started this project, I would say 8 years ago, maybe 7 years ago, and we decided to look at components of fluid ionic balance machinery. And then Mehdi Jenkins joined the lab, and both of them, I think they made a significant contribution to our understanding how fluid ionic balance works during hibernation. And this is going to be a topic of my talk today. So fluid balance is essential for survival. We all know that we need to drink water, and even overnight water deprivation, in our case, will cause dehydration. So we're constantly keeping homeostatic set point in our in our body. And in case of blood components, our blood osmolarity is 290, 300 milliosmoles. This is the optimal set point for us. And a little deviation from the set point, as little as 1%, will cause thirst. We meaning our body, we constantly defend our set point. We monitor deviation by our specific, by brain regions that are specific for feeling changes in homeostasis, and we're also making corrective and anticipatory action. What it means that if we're dehydrated, we're feeling thirst and we start to drink water. And also when we're dehydrated, we start to retain water. And kidney, they are very important for regulation of this process. So we have two arms, behavioral arms such as thirst, and we have water retention such as hormonal arm. Both of them, they work together in order for us to maintain homeostatic set point. So for how long different animals can survive without water? So in 1959, it has been shown that antelope, ground squirrel, these animals, they're close relatives to our ground squirrels, but they don't hibernate, they live in desert. And in this publication, it has been shown that these animals can survive without water for 9 days, which is also actually pretty good. So in our case, we will not be able to survive in desert without water for 9 days. Usually when we talk about dehydration, the first animal that comes to my mind is actually camel. And there is a literature on camels as well. In 1956, it has been shown that camels, they can withstand prolonged period of dehydration up to 60 days. But if you look on average, usually these are 2-3 weeks when camels can survive without water. So in our case, our ground squirrels, they can survive without water during hibernation for up to 7-8 months. So they are in fact champions for survival of dehydration. So how fluid homeostasis is tweaked to serve the needs of hibernation? And this is really oversimplified diagram how fluid ionic balance works in our body. So there are specific subset of neurons that are located in subfornical organ. This brain region, SFO, doesn't have a blood-brain barrier. It has an access to the blood directly, and it can feel actually what is happening in the blood. And if we eat something salty or if we don't drink water for some period of time, concentration of osmolytes in the blood will increases, and SFO, subfornical organ, will detect it. And it will immediately send signal to two brain regions, one brain region responsible for thirst regulation, and we start to drink water. Another brain region is responsible for release and production of antidiuretic hormones. In case of rodents, these are vasopressin and oxytocin, and they're produced in hypothalamus. So during dehydration, we will drink water, we will release vasopressin, oxytocin in the circulation. Both of these hormones, they can bind to these receptors in kidney, and kidney will retain water. Together with drinking water and retaining water, we are bringing our set point to the normal level, and then blood concentration will be, in case of mice and rats or us, it will be 300 milliosmoles, and the SFO will tell the rest of the brain, we're fine, we're not going to die. So when we started this project, we decided to look at every component of this machinery. So we looked at the blood osmolarity, hormonal level. We look at the behavior. We asked, are they thirsty actually during hibernation? And we also look at the brain activity, specifically brain regions that are responsible for thirst regulation and release of antidiuretic hormones. So again, in face of water deprivation, what do we know? What's happening with animals? So in our case, mammals, we increase vasopressin in preparation for sleep. Vasopressin is very tightly regulated by daylight cycle. And before going into bed, we have a huge spike in vasopressin in the circulation, because our body will try to prevent water during sleep, because if we feel thirsty, if we're dehydrated during sleep, we will wake up and drink water. In this case, this will actually disrupt our sleep pattern. That's why we usually release more than we need in the circulation to maintain fluid ionic balance during sleep. In case of camels, they hugely increase plasma osmolarity during hibernation, during dehydration, and they also have a huge increase in vasopressin. So both of these components are usually goes together: increase in blood osmolarity and increase in antidiuretic hormones. In case of desert rodents, the same happening: increase in blood osmolarity, increase in vasopressin. Also, renal aquaporins, they have, they're going up to the roof in case of desert rodents. So when we started this project, we actually was thinking that our ground squirrels will have increase in thirst, and our ground squirrels will have increase in vasopressin. So they will retain water, and they also will be very thirsty during hibernation. So again, our prediction will be osmolarity will go up, vasopressin and oxytocin will rise during hibernation. However, when we measured it, what we found that uh, osmolarity during interbout arousal is identical to active animals. And here you have to remember that active animals, they have ad libitum of water, so they can drink pretty much any time. In case of these animals, they didn't see water for several months, and the blood osmolarity is absolutely normal. What was also puzzling that during torpor, they have decrease in blood osmolarity from 300 milliosmoles to 300. So it's significant decrease. It's around 10% decrease in osmolarity. We're currently trying to figure out how do they do it, how they're going back and forth. And this is a topic of the current investigation. So we were wrong in terms of our prediction. Blood osmolarity, absolutely normal. The same actually with vasopressin. So if you look with the vasopressin level, despite that they didn't see or didn't drink water for a long period of time, level of vasopressin is identical to active animals, and the same for oxytocin. So it seems like they have ability to maintain normal blood osmolarity, normal level of antidiuretic hormones such as vasopressin and oxytocin, despite that they actually don't have access to water for several months. Next question we asked, will they drink water? So if we give them water in the middle of IBA, when animals are 37 degrees Celsius, will they drink it? And for that, Nifeng, she developed a behavioral assay. It was never done in hibernating species before. And we can actually score precisely level of drinking in ground squirrels. And here you can actually see how ground squirrels drink water. So when we performed behavioral experiment, we were shocked. So during interbout arousal, if you give animals water, they are not interested in drinking water at all compared to active animals. And again, these animals, they didn't drink water, they didn't see water for several months, and they are not interested in it. So what it means that they have a very potent inhibition of thirst during hibernation. And what are the speculations? Why do they need it? If you remember, so our animals, they wake up every 2-3 weeks and they go through IBA. And if they sit in underground burrow and they feel thirsty every time when they wake up for IBA, they will search for water. They don't have water in the underground burrow. They will escape from safety of their again underground burrow, and this will increase risk of predation, and this will change hibernation pattern and potentially may disrupt hibernation maintenance. So very potent inhibition of thirst is actually, we think, is important for maintenance of hibernation. So what is happening here, and this is very important for the rest of my talk. Again, we have two arms in our body. Behavioral arm, when we dehydrated, we increase thirst level and we drink water. And hormonal arm, when we have increase in vasopressin for water retention. These two arms in our body or in any mammal, they're all coupled. You cannot have one without another. In our case, what we found that behavioral arm is significantly reduced. Basically, behavioral arm thirst is suppressed, and hormonal arm is fully active. And this is, again, the first demonstration in any mammal that behavioral and hormonal arms of fluid homeostasis are decoupled during hibernation. And again, this is very important because they need to have vasopressin and oxytocin in the circulation. Because if we don't have vasopressin or oxytocin in the circulation during IBA, they will actually pee to death. Because patients with, we know that if you block signaling, peripheral signaling of vasopressin, and you block receptors in the kidney for vasopressin and oxytocin, this will cause severe diuresis. So to keep hormonal arm is very important. And we ask the question, how do they actually decouple these two arms, behavioral and hormonal? Uh, Again, just to remind you that subfornical organ, SFO, is very important for regulation of thirst and is also very important for sending signal to this arm to release and produce vasopressin and oxytocin. And we hypothesized that this arm, hormonal arm, is fully active and thirst pathways are inhibited. So we decided to check this hypothesis, and first we started with hormonal arm. Before we go into actual data, I want to show you our recordings and show you how animals look like when the body temperature is 4 degrees, when the body temperature is 10 degrees, and when the body temperature is 27 degrees Celsius. These time points are very important because we sampled blood during torpor arousal cycle at 4 degree, 10 degree, 27 degree body temperature. So we all know who are working on hibernation that during torpor, they're very immobile. At 10 degrees Celsius, you already start to see some movement because they switch on shivering, non-shivering thermogenesis. And at 27 degrees, they're fully active. So we performed measurements of blood osmolarity during arousal, and you can see that it takes usually from 1 hour 30 minutes up to sometimes 3 hours. And when we measure blood osmolarity through transition, we see actual gradual increase. So already at 10 and 27, there is increase compared to torpor, it's slight increase, but this increase actually will be very significant at the physiological level. So we think that they have a gradual increase through this transition during torpor arousal cycle. What was actually very exciting for us to see that in case of vasopressin, we see that they actually reaching level almost the same as active level when the body temperature as low as 10 degrees Celsius. And at 27, they are already at the level of active animals and the level of IBA. The same is happening with oxytocin. What it means that hormone release is underway as at 10 degrees during torpor arousal transition. At least we see it in the blood. You cannot release vasopressin and oxytocin without activity of neurons that are responsible for release of these hormones. And we decided actually to look at activity of these specific neurons using fiber photometry. So what we did is we injected our ground squirrels in the hypothalamus, specifically in the brain region called SON, that is responsible for production and release of vasopressin and oxytocin. We use CAMK2 promoter to label excitatory neurons, and this brain region is primarily in excitatory neurons. There are primarily two populations, only vasopressin and oxytocin. Then we've used this with GCAM6, and this is a calcium indicator. And when there is increase in neuronal activity, you will have increase in delta fluorescence. We packaged it into virus, AAV virus, specifically serotype 9. Again, we injected during summer. We also injected fiber in the same brain region in order to perform in vivo imaging during torpor arousal cycle. Then we have to wait for around 6 weeks. That's the minimum. And then we hibernated these animals and we perform fiber photometry recordings through the whole torpor arousal cycle. And usually it takes up to 3 hours. And what we found that neuronal activity we started to see neuronal activity as low as 10 degrees Celsius. So you see that the baseline increases significantly already at 10 degrees. What it means that the hypothalamic neurons in SON, they are very active already. But again, these are mixed bag, and we don't know because CAMK2 may labeled also other excitatory neurons. So what we decided to do is we decided to stimulate these animals with a very potent stimulant, 2 molar of sodium chloride and 3 molar of sodium chloride. This is a standard way to stimulate this type of neurons because this injection will cause increase in blood osmolarity by 30, sometimes 50 milliosmol, and you immediately engage in release of vasopressin and oxytocin, and you immediately activate SON in mice, rats, or also in humans. So we decided to inject torpid animals when they still at 4 degrees Celsius and see when the system will start to feel these changes in blood osmolarity. And we were actually very surprised and pleasantly surprised. What we found that already at 4-5 degrees Celsius, when they're very, very cold, body temperature is very cold, the SON neurons, they actually can feel it and they increase activity compared to control. We also performed fiber photometry recordings in control group in brain region adjacent to SON, but this brain region has nothing to do with fluid ionic balance. And we actually see that these neurons are silent. So these experiments are very exciting for us for several reasons. First, that we can employ fiber photometry in hibernating species. Second, that we can perform activity and we can see robust signal at low body temperature at 4-5 degrees Celsius. And third, from scientific point of view, we demonstrated that these neurons are active at 4-5 degrees, which is actually for mammals is unheard of because the whole synaptic transmission in mammals are blocked below 15 degrees Celsius because our electrogenic machinery cannot operate at 4-5C. So here we demonstrated that osmosensitive neurons can be activated at extreme low temperature. And also what we demonstrated that vasopressin oxytocin pathways, they are tuned to actually act as early as 4-5 degrees Celsius. And that's when they, during initial phase of arousal, when they immediately started to release vasopressin and oxytocin, so to retain water during torpor arousal cycle. And then they completely online, they reach the level of full activity, and vasopressin and oxytocin reaches the level of active or IBA animals at the temperature as low as 27 degrees Celsius. So they already at the level of active. So this pathway is active. What's happening with thirst pathway? And here I want to introduce more players. This is very important because it's not only about vasopressin and oxytocin. So our kidney, when they are dehydrated, when they feel that we're dehydrated or we're fluid deficient, they also release angiotensin and aldosterone. These are two hormones that are go back to back with our fluid ionic balance. They're very important for the regulation. And angiotensin specifically, when there is increase in angiotensin, it means that we're very thirsty. So angiotensin will act specifically on lamina terminalis, subset of neurons in SFO. And here we're also going to talk about OVLT. This is a brain region that is also important for regulation of thirst. So OVLT and vasopressin and SFO, they have receptors specifically for angiotensin. And if we dehydrate it, we will have increase in angiotensin, and this increase in angiotensin will produce thirst. So we decided to look at both hormones, angiotensin and aldosterone. And what we actually found that during hibernation, during IBA, there is a significant increase in angiotensin and there is a significant increase in aldosterone. So what this is telling us that kidney actually, they feel dehydration, but brain by some reason doesn't. So kidney perfectly capable, responsive to not seeing water for a long period of time. And that's what actually happening in our body during dehydration and also when we're losing volume. So when we're losing volume, we become very thirsty, but also we started to crave for salt. Uh, as a result, there is increase in aldosterone. So we hypothesize that by looking at it, that they actually should crave for salt with such a huge increase in aldosterone. We perform experiment again, and this is not surprising. This is cumulative drinking. If we do experiments in active animals, and here we give them zero point 0.5 molar of sodium chloride, which is actually concentration of seawater. This is aversive for us and for most of the rodents. They don't drink it. They just drink water. In case of hibernating animals, they actually shifts it. So they are not interested in regular water, but they're very interested in salty water. And they even started kind of craving. They have this potentiation. More they drink, more they want it. So this is consistent with our results in terms of increase in aldosterone, so the craving for water, but together with increase in angiotensin, what this is telling us that our squirrels are fluid deficient during hibernation. Their brain just cannot interpret it. So we decided to look at the components, neuronal components of fluid ionic balance. Again, we looked at SFO and we look at OVLT. These two brain regions, they don't have a blood-brain barrier and they directly can sense angiotensin and also aldosterone and then signal downstream in the brain. So first we ask the question, maybe our ground squirrels, they cannot feel angiotensin. Maybe they don't have receptors. Maybe they have downregulation of these receptors. And we perform analysis when we can label receptors with a specific dye. And here you can see fluorescence where the entire subfornical organ, it has this oval shape. And we perform this fluorescent labeling, so we can see that angiotensin receptors are present during IBA and during active state. We also perform measurements of these receptors at the level of RNA, and we didn't see any difference between active and IBA state. This is telling us that receptors are present, but one thing is actually to see fluorescence, and another thing is actually to test functionally. So how we can test functionally that these neurons are sensitive to angiotensin? And what we did is we took neurons from subfornical organ and from OVLT, two brain regions that are responsible for detection of angiotensin and for initiation of thirst. We put them on a dish, and then we performed ratiometric calcium imaging from these neurons. And what we did is we applied 10 nanomolar angiotensin. This is physiological concentration for both preps. And at the end of experiments, we also apply high K. This is our kind of double control to make sure that we're recording from neurons, because only cells will become fluorescent after high K application. This will be neuronal cells due to depolarization. And then we scored amplitude of the responses for angiotensin in two different brain regions. And what we found that in case of SFO, active and IBA animals, they have the same sensitivity at the neuronal level to angiotensin. In OVLT, we even see increase in sensitivity during hibernation, which is actually opposite to what you would expect when you don't have thirst. We also calculated percentage of the responsive cells. And again, we saw opposite from what we would expect. We found that during IBA, there is increase in number of angiotensin positive cells that could detect angiotensin from the blood compared to active state in two brain regions. So it seems like that those neurons can sense angiotensin during hibernation. Next, in parallel with that, Mandy performed serial reconstruction of subfornical organ and OVLT, SFO and OVLT through the whole volume. And one dot here represents one animal, and usually you need to reconstruct six or seven sections, 40 microns thickness through the whole volume. And then we calculated number of C4 positive cells. And C4, this is a proxy for neuronal activity. More C4 cells you have, it means that you have more activity in that particular brain region. And what we found that there is a significant decrease in number of C4 positive cells in SFO and OVLT during hibernation. It means that these brain regions are less active or are suppressed during hibernation. So what's happening actually there? We decided also to perform tests when we can artificially activate these neurons at the physiological level. So what we did is we inject ground squirrels active NIBA with 3 molar of sodium chloride. We can increase the blood osmolarity, normal blood osmolarity at this level, 330. And then we, after injection, we can increase it to 365, 370 in both cases. What will happen in our body and in mice or rats, you immediately will have increase in number of C4 positive cells due to increase in activity in this brain region. And that's what we actually saw in active animals. But during IBA, we didn't detect any potentiation. And the same in 2 brain regions, SFO and VLT. That means to us that those neurons are less sensitive to osmolarity changes during hibernation. And this is consistent with our previous C4 staining when we can look at this just steady state without any activation. So we decided to look at intrinsic properties of the cells. We thought that perhaps there is something is happening with electrogenic machinery, and these changes are happening seasonally. So we perform electrophysiological recordings in active animals and during hibernation from subfornical organ neurons. And here you can see that in case of active state, we see perfect spontaneous spiking activity. The same you would see in this brain region in mice and rats, and it has been demonstrated by other groups and other rodents. However, when we look at the animals during hibernation, during interbout arousal, we actually saw a significant decrease in spiking activity, and you can see the striking phenotype even visually. So they can spike, but they actually don't perform well compared to active animals. We also look at the electrophysiological properties of those neurons during hibernation. So we look actually first at the cell diameter. This is our internal control, make sure that we're recording from the same cells. We look at the input resistance. Input resistance is significantly affected. We have huge increase. We also see significant hyperpolarization of neurons during hibernation. And people who are working on electrophysiology, you appreciate that 20 millivolt shift in terms of resting membrane potential is huge. Usually in clinics during pathological conditions, 5 millivolt shift will cause complete elimination of pain sensitivity at the level of somatosensory peripheral nerve fibers. In this case, we're talking about 20 millivolts. So it's actually significant, and this would explain why these neurons are less active. So it's just harder to activate them. And as a result, we see decrease in spontaneous firing rate. So this is quantification of the previous slide. We also analyzed in details different parameters of action potential, and at every parameter we actually see difference that pointed us that action potentials, even they can produce it, these action potential parameters are not as good as action potentials during active state. So we look at the time to peak, we look at the peak amplitude, and we actually see in case of time, they're much slower, and peak amplitude is actually decreased, as you can see from this graph. We also next level. We also perform RNA-seq of subfornical organ, and we look at different genes that are up or down regulated, and one of the molecule was on top of our list was GABA receptor. And GABA, these are inhibitory molecule. If you have more GABA synaptic transmission, it means that you have more inhibition. And GABA receptor was on top of the list as a fornical organ during hibernation. And we thought that this increase may cause changes in the properties of these neurons with regard to susceptibility to inhibition. And what Mehdi did, she performed experiments when she first just recorded from SFO neurons in active state. Then she applied GABA, and you immediately see reduction in the firing rate. And then she washed GABA, and she can see that firing rate goes back. In case of IBA animals, to begin with, they have decreased baseline, but then if you apply GABA, you're basically blocking all firing. And then when you try to wash this, you cannot. It means that you have probably different affinity to GABA during IBA, and we explain it perhaps due to increase in GABA receptors. We're also trying to understand now actually what's happening here, what are the molecular underpinning for that. But what we know that neurons are they have more susceptibility to GABA inhibition during hibernation. So together, what I showed you so far, that kidney, they can feel that animals are dehydrated, that they fluid deficient. What I also showed you that during hibernation, subfornical neurons, they change properties of the electrogenic machinery, so they cannot fire as efficient. So they're healthy, but they just don't want to fire. And this changes due to seasonality. And what we also think that uh, these neurons are more susceptible to inhibition by GABA. And currently we're checking that there is an input from other brain region that is actually GABAergic to SFO neurons. And we think that that brain region is more active, and it's actually releasing GABA and tonically inhibits seasonally cells during hibernation. So how to survive for 8 months without water? Again, we just at the beginning, and this is a foundation for the future studies. We think that vasopressin and oxytocin neurons, they remain functional at lower temperature, and this allows them to release antidiuretic hormones in the circulation during torpor arousal cycle. And this is one of the important things for to stay hydrated. And we also demonstrated that those neurons, they remain hypoactive during hibernation, and this allows them to suppress thirst throughout the entire hibernation. And we also speculate that this is very important for hibernation maintenance. You don't want to be thirsty in the middle of hibernation. So with that, I would like to acknowledge my group and group of my close colleague Slav Bagriantsev. We work together. And specifically, I would like to acknowledge two superstars, Nifeng, she has her own squirrel lab in Wesleyan University, and she actually initiated this project in my lab. And develop a lot of tools and assay. And also I would like to acknowledge Mehdi Jenkins, former graduate student. She just left a month ago, and she developed electrophysiological recordings, also was involved in fiber photometry, and both of these ladies, they're absolutely fantastic. I also would like to acknowledge our collaborator, Dana Merriman. Without her continued support, this line of research would not be possible. And I would like also to acknowledge our finding. Our lab is curiosity-based lab, and we really appreciate support from all these foundations. Thank you very much for your attention.
Thank you very much. That was a really nice talk. Excellent body of work that you put together on this subject. So we have some questions. I think I just saw Claudia's hand was raised. We go with that first. And
sorry, I just wanted to applaud.
Oh, okay, no problem. Yeah. So then maybe we go to Jim.
Thank you very much.
Thanks, Shauna, and thanks, Elena. Great talk. And I wanted to welcome everybody to International Squirrel Appreciation Day, which we all know happens on the 21st of January. But Elena, my question was related to the in situ responses of the neurons in the brain when you activated the mode. Now, you measured body temperature using telemeters in the abdomen, is that correct?
So for that particular studies, we measure them here. So we measure them, they deeper than subcutaneous, but we use BDMS. So this body temperature, I would say they were underestimated. So they were warmer than in abdomen.
Well, that's interesting, because I was going to say that there's a significant heterothermy between the abdomen and the brain
Mhm.
heats up more quickly. So if
Yep.
they were just interscapular, then
Yep.
it might actually be overestimating, because that's the bit that warms
Yeah,
up faster. But
they were overestimating. Sorry. Yeah.
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 Okay.
Yep. Mhm.
Okay, thank you. That clarifies things.
Okay. In the meantime, we'll wait for anyone else who wants to put their hand up and ask a question. I will. Oh, well, we'll just go. I'll let someone else ask rather than ask my own question yet. So, Catherine, would you like to go ahead? We can't hear you. Catherine, would you like to ask a question?
Sorry, it took me a second to find the switch. Hi, it's great to see everyone. You can see me too in my, no, you can. Now you can in my messy office. I have one question I'm always wondering. So you said if we put you into a fridge, you die, become hypothermic and die. How about the squirrels in summer? So I would like to know your opinion on how could they do it spontaneously? How long? What kind of preparation do they need? Or if you put the summer squirrels into the fridge, would they still do that, or would they also just die?
There is a literature on that actually. So you cannot induce hibernation in June. So then very strong seasonality. We did this experiment with our animals during summer, so they don't die. So they have adaptation at the genomic level. So with regard to temperature, we think that they have already, so it's kind of in their genome, so they're cold resistant compared to us. But on top of it, they also have some seasonal changes in electrogenic machinery. In the peripheral nervous system, we don't know about central. I would expect that in central as well, that even fine-tune that electrogenic machinery to for prolonged cold.
Okay, thanks. So, but you have, so your summer squirrels, you had to put into cold temperature for a while before they. No?
And they fine. Yep, they don't die.
That's good.
Okay, thank you. Anusha, would you like to ask a question?
Hi, I'm in a noisy place. Can you hear me okay?
Yep.
Yes.
Hello. Mm-hmm.
Okay. I study hummingbirds, and I've always been curious if you can like suspend the brain of a hibernating squirrel, would it still be hibernating? Like, can you take cells or tissues or something from a hibernating animal, and would they still be able to hibernate, or do they need the context of the organism?
So you're asking about intrinsic properties versus properties that are I think both. In the laboratory of Wei Li, there was a publication in Cell several years ago. They actually demonstrated intrinsic properties at the level of mitochondria proteins, and that makes them resistant to cold. Although I would say that at the level of the organism, you also need to orchestrate a lot of functions during cold, and I would say that it's a combination. But you need to have also intrinsic properties of the cells in order to withstand prolonged period of cold. If we're talking about cold, in terms of metabolic adaptation, I actually don't know.
We seem to have a comment here from Jim, but perhaps you should just go ahead and say it.
Sure, if there's time. Anusha, it's a great question. We've isolated hepatocytes from hibernating ground squirrels, and they do not retain the metabolic phenotype of the whole animal. So if you if in order to do that, you have to perfuse them at 37 degrees for a couple of hours, and then condition them. And even if you make them cold after that, they seem to be exactly the same as summer cells from summer animals. So, like Elena said, there's some things are intrinsic and some things are organism dependent.
Okay. Perhaps I can get a chance to ask my question. So I'm actually quite interested in the pre-hibernation state, which you mentioned at the beginning of your talk, and
Mhm.
I was wondering if you'd looked and seen any changes in thirst or neuronal responsiveness in that state, or whether you'd
Yes,
looked.
we did, and part of it is published already. So we looked at pre-torpor state, and even during this state, they have reduction in thirst. So they already have suppressions. It's a
Okay.
seasonal. So I would say that in pre-torpor is not as dramatic as during IBA. But I think that all prerequisites, neuronal or whatever connectivity, this is already happening in preparation for hibernation.
Okay.
And in terms of blood osmolarity, we do see that it's intermediate. So we see decrease during torpor, right? And in pre-torpor, it's intermediate between active and torpor. So again, this is telling us that there is a significant component of seasonality present.
Okay. Yeah. Thank you for that. If anyone else has a question, just go ahead and raise your hand. Okay, Roloff?
Yeah, thanks, Elena. It was a wonderful talk.
Thank
You compared in the beginning to camels.
you. Mhm,
Camels get their water from burning the fat, hence the humps, right?
mhm,
Would
mhm.
you go as far as saying that the ground squirrels use the arousals where they burn a lot of fat to provide the water?
Mhm,
Because you may not be thirsty, but you still need water, right?
mhm, mhm.
Is that a crazy way of putting things? What would you say about that?
Uh, so you're talking about metabolic water. So we
Yeah.
perform this quantification, how much you actually during the entire hibernation you need to burn in order to stay hydrated at the 30 milliosmol, which is the set point. And they need the body weight of ground squirrel. And I can, if you're interested, I can send you actually this quantification and more details. So body weight of our ground squirrels is around 275 grams. In order to stay hydrated and use only metabolic water, they need to burn 300 grams of fat, more than the entire body weight of squirrel.
Yeah, but then you are assuming a certain ventilation rate, evaporation rate, which of course is also greatly reduced. So it's a bit of a difficult calculation, right? Because are you assuming the evaporation rate being high only in the arousals? Is that what you're saying?
No, what I mean is we calculated, so every torpor arousal cycle, they're going from 300 milliosmol to 330 milliosmol, which is 30 milliosmol per IBA. So in order to go for 30 milliosmol, you need basically every IBA, roughly you need to burn 10, 15 grams of fat. Just to go for this difference. We know that this difference is happening. So if you have 20, 25 IBAs and they're burning 15 grams of fat to go through this to stay hydrated, it means that 115 by 20 is 300 grams. So basically, we think that metabolic water is important. And we know that they burn fat, and we know that they have a huge increase in beta hydroxybutyrate during hibernation and during IBA. So this is primarily their food source. But we don't think that metabolic water, this is the only thing that actually contributes to fluid ionic balance. We think that something else is happening, but we just don't know what.
But at least it's part of the story. Yeah. Okay.
Yeah, exactly.
Thanks. Thanks.
And actually, in case of camels, it's interesting, and we also checked it on ground squirrels, because camels, it's not only burning fat, they have anticipatory drinking. So they know that they constantly anticipate that they will be dehydrated, and when they have water, they drink 5 liters of water, and they store these 5 liters in the stomach, and they have special organization of stomach. And then when they go into deserts, they partially release this water. Very smart. So this to stay hydrated.
Okay, thank you.
Okay. Do we have any other questions? I think we're pretty much getting to the end of our time now. But we have someone with a microphone with issues. Maybe I'll have a look at the message in the chat. So this is from Cord Brunage. If I heard correctly, the ground squirrels were actually hypoosmotic during torpor. Do you think that the kidneys are responding to changes in blood pressure or natriuria?
Okay, so during this is actually related to the previous question. Yeah, so our ground squirrels during torpor, they change the blood osmolarity from 330 to 300, so they hyperosmotic compared to active or IBA state. So we think that kidney significantly contributes to this, yes, because they're constantly monitoring what's happening. But we don't know pathways, so we didn't check pathways at the level of kidney yet. But I can just only speculate what's happening, but we think that this is important. It's not only brain that is involved in the regulation. They orchestrated together.
Okay, thank you very much. And Cole has raised his hand. Would you like to go ahead and ask your question, Cole?
Yeah, thanks. Great talk, Elena. I am curious your thoughts on you're saying that these animals need to defend around 330 osmolarity. How about the possibility of, a sliding set point across hibernation? Is it 330 that they're defending against across the course of the year? Or do you think there's a possibility that that's actually changing and sliding around?
Okay, that's a very good point. What it mean? So what Cole asked us is, do they have a different set point? If they have a different set point, like let's say if neurons changing the set point to 360, then you kind of don't need to drink because you feel that you're constantly hydrated, right? That's what you're asking.
Yeah, totally.
This is definitely possible. The argument against that at the level of subfornical neurons, if we didn't detect change in electrogenic machinery, I would say that this may be a possibility. But because we do see massive hyperpolarization and changing in the properties of action potential, then I would say that a major contributor is probably seasonal changes in electrogenic machinery. But what you're saying, this is not mutually exclusive. They may be complementary. And it actually would make sense to test it, but it's hard to test because you just apply different osmolarity and this will produce a lot of non-specific effects.
Hello, everybody.
Hello.
Hey. Would you like to ask a question, Erhard?
Oh, I think we never met you. Nice to see you on the screen. No, I don't want to disturb the discussion, which is obviously ongoing at the moment. Are you presenting a talk or what are you doing?
I'm already done.
Yeah, the session started an hour ago. We've already had the talk. Hopefully we've managed to record it. We'll see.
Oh, then I missed it. I'm sorry.
It's nice of you to try to turn up. So good. Yes, so we were just wrapping up actually. So if there are no other questions, Roloff, yep, go ahead.
Yeah, just to say that I'm really enthusiastically see so many people coming up to these seminars. I counted 64 people at the peak. Wonderful. Please consider. I put it in the text box. Please consider, if you're not already a member of the Hibernation Society, please consider becoming a member so that we can continue to have these lectures So I put the link hipsoc.com in the text box, so you can find it and maybe become a member. Thank you all for being here.
Okay, great. Well, thank you very much, everybody. Hopefully, we'll run some more of these sessions soon at a different time because of our very international audience. So we'll be stretching around at times to make it more convenient for different parts of the world. Yeah, so thanks again, and
Thank you very
thank you
much.
very much.